Thank you to everybody for attending today, where I'm going to tell you a story that I spent a few years working on while I was in a postdoc at the NIH. So the majority of this work comes out of the NIH and has actually been finished here while at Central. It's currently in revision and should hopefully be published, fingers crossed, in the next few weeks. So if you want more information on this, hopefully in a few weeks I can send you the manuscript that is published. So what I'm going to really be talking about today is a story, and it's a mouse story is the way I think about it. This story is going to involve talking about inflammation and NSAIDs, the use of ibuprofen drugs, which act as non-steroidal anti-inflammatory drugs, or NSAIDs is how we get that name. Most of us know it as ibuprofen or Advil, which is easier to say than NSAIDs. We're going to talk about a mouse called Nag, and I love this name for this mouse. So you're going to hear me talk a lot about Nag. These mice kind of do nag a little bit because they don't have proper lactation or mammary gland development, which will bring us into talking about how the mammary gland develops, and then some conclusions that we can make from this. So first, why study inflammation and NSAIDs? Inflammation is associated with a variety of different diseases and disorders. Not only do we get inflammation if we fall down the stairs and sprain our ankle or twist our arm, we also see inflammation in the case of allergies. We see autoimmune diseases are predominantly due to excess inflammation. Pulmonary diseases, the cardiovascular system can be affected by inflammation, which alters its ability to function normally. A variety of different diseases that we see here. So that brings us to think about how does inflammation work and how does the inflammatory process actually work? So what's happening is we have capillaries where blood cells are moving through them. This is spread throughout our body. We've got our red blood cells carrying oxygen out to many different areas. There are also some white blood cells or immune cells that are present. If there is a trauma or some type of event that will induce inflammation, let's say we fell down the stairs and twisted our ankle. I use that as an example because I may have done it once or twice in my life. What happens is after that insult or injury, we get a response called capillary widening, where the capillaries get wider. They actually get increased blood flow to that area where there is this inflammation taking place. This can also happen if you get a cut on your arm. You get an inflammatory response. With that, we get the normal symptoms that we think of as inflammation. You get increased heat in the area. It turns red. It's warm to the touch. We get swelling. It can be associated with pain. So this is all happening in the capillary level from these red blood cells and white blood cells. We also get an increase of those white blood cells or those immune cells into the region that's hurt. And these cells are acting to correct the damage that might have taken place to the cells in the tissue. I use here white blood cells. Those of you that have taken immunology or will be taking immunology next quarter when it's offered will get much more detail on this inflammatory response. But for today, we're going to think about these white blood cells coming in. What they're going to be doing is actually releasing some enzymes called prostaglandins. So you get an increase in prostaglandins within that area, and that's part of what's causing that inflammatory response. So how do NSAIDs work, or why do we take ibuprofen? NSAIDs are non-steroidal anti-inflammatory drugs. I'm sure we've all taken some at some point in our lives. The whole goal of these is to reduce that inflammatory response, reduce the inflammation that's occurring. We've got a variety of over-the-counter drugs as shown here, as well as several different prescription medications that can take place. And all of these you're taking so that you can reduce that inflammatory response that's happening at the level of the capillary. So if we go back to our model of how NSAIDs are working, with this increased response, I mentioned that we get prostaglandins. We're not going to focus on what prostaglandins are, but know that they're happening in response to this inflammation. And they're actually made by enzymes that are called COX enzymes. So there's COX-1 or COX-2. These are two different proteins. And the whole job of these proteins is to increase the prostaglandins. and that's going to help with that immune response. So how do NSAIDs work? How are they actually working to reduce that inflammation? They're working by blocking the activity of these COX enzymes. If we block the activity of those COX enzymes, we no longer get the production of those prostaglandins. That, in turn, is going to reduce the inflammatory response within the cells. That's going to bring down the level of swelling and help to heal the tissue. So the whole goal of these NSAIDs is to reduce the activity of these enzymes. That's why you can take them over time. After you, let's say you take an ibuprofen, a few hours later, you're going to start to get those symptoms back. That's because they're no longer inhibiting that enzyme. You're building up those prostaglandins, which is going to lead to the second dose that you may take later on. So in the early 2000s, there was some researchers that were at the NIH that wanted to understand how is it that these NSAIDs are working? What are they actually doing to the cell? So what the researchers did was take a dish of cells, these are cells grown in culture, and they induced the inflammatory response in these cells. So they made these cells think that there was some type of injury. And they then took NSAIDs, crushed up all of those different types of pills that we saw, and treated the cells with them, and got reduced prostaglandin production. They reduced that inflammation level, and they wanted to understand what type of mechanism might be taking place. So they did an experiment called a microarray analysis. And I'm not going to go into details for this experiment, but what it does is it allows the researchers to look at over 55,000 different probes. This is looking at all of the genes that might be changed when you treat with drug versus when you don't treat with a drug. So essentially the question that the researchers are asking is, what does that drug do to those cells? And what they found was a significant increase in a gene called NAG1. NAG1 stands for NSAID activated gene 1. It's a very creative name. That gene goes up after you take an NSAID. So if you take ibuprofen, you get higher levels of NAG1 in your bloodstream. This has been shown in humans. A lot of the trials have been done in males, where if males are taking ibuprofen, they can actually see an increase of NAG1 in the bloodstream. NAG1 is a TGF-beta family member. I mention this because I know a few of you are taking cell biology or developmental biology and have heard of this before. We talk about these proteins. They are secreted into the bloodstream. So rather than acting in just one location within the body, they're actually getting into the blood and circulating throughout our entire tissue. So after we take NSAIDs, such as ibuprofen, we get an increase of NAG1 within our body. And the researchers hypothesized that this NAG1 might be causing a difference. So the next experiment that they wanted to do was look at, do all NSAIDs do this? Do only, say, ibuprofen or Advil? What about some of these other types of drugs? And so what they did is they again took cells and treated them with a variety of different drugs, such as NSAID, and then looked at whether NAG1 was induced, if they could see that NAG1 expression. And what I have here is a list of a variety of different NSAIDs. There's aspirin, there's our standard ibuprofen, acetaminophen, as well as some specific inhibitors. And the one thing that they found is that no matter which drug they were using, which NSAID they were using, they saw an increase of NAG1 within those cells. So those cells were all making increased NAG1. They were all secreting that protein out so that it could act. So from this, the researchers came to the hypothesis that NAG1, NSAID activated gene 1, might contribute to a reduced inflammatory response. It goes up right after you take NSAIDs. The reason we're taking these NSAIDs is so that we can reduce the inflammation. So it makes sense that these drugs might be helping to reduce the inflammation that we're seeing. But all of these experiments were done with cells, in a dish, in a lab. I mentioned that there's studies in humans that have shown that NAG1 goes up, but nobody's actually looked at how does NAG1 act in vivo, or ask the question, what is the role of this human gene in a living organism? It's really hard for us to go collect a whole bunch of people and say, hey, come here, can we give you a bunch of these NSAIDs and then take different tissues from you and see what's happening? You can't do that, right? Those experiments are not done anymore. So what the researchers did was they made a mouse called Nag. This mouse expresses the human gene NAG1. So it expresses human NAG1 that usually goes up after you take NSAIDs. So for the remainder of the talk today, we're going to be looking at experiments using a control mouse, which has no NAG1 expression, and using the NAG mouse, which is expressing the human NAG1 gene. So what they did is they took DNA for NAG1 from the human and added it into this mouse. So it's essentially like this mouse is taking ibuprofen every 4 hours, all day, every day. Okay, high level. That doesn't happen in humans, right? Very rarely do we take it consistently long term, although there may be times when that isn't the case. Say when we fall down the stairs and hurt ourselves, we are taking it more often than we need to. What they found is that NAG1 is secreted into the bloodstream of these mice. So when you go into the doctor's office, one of the things that they'll do if you're doing a physical is they'll take a blood sample from you, and they'll test it for different hormones, for different markers that are a readout of how healthy you may be. If we take some blood from the NAG mouse, what we see is increased NAG1. So it's made its way into its bloodstream and it's circulating through these animals. So the researchers have this NAG mouse, and the question becomes, what type of experiments should we test? What do we want to see that happens? There are a variety of different diseases and reasons that people might have increased inflammation. And due to the current environment, where our population is undergoing this increase in body mass and there's increased obesity across our country, the researchers thought, let's look at obesity. So obesity is associated with increased inflammation. The fat or the adipose tissue of people that have higher BMIs has more of those inflammatory responses within them. So this shown here is a mouse, a control mouse, and then this is the leptin knockout mouse, which is originally used as a model of obesity. You can see it's much heavier than the control mouse. I've actually seen some mice that are about this big, and it's really hard for them to walk around their little cages because their little bellies are bigger than they should be. And so the researchers that make the NAG mouse wanted to know, how do these mice respond to increased obesity? What happens if we put them on a high-fat diet? So the researchers took our control mice, they took the NAG mouse, and they gave them what I call the Big Mac diet. So they gave them food that was over 60% fat. Almost all they were eating was fat. In a normal mouse, if they're eating this Big Mac diet every day, they get fat. Makes sense, right? Not good to be eating a Big Mac diet every day. It's not healthy for us. What do you think might have happened to the NAG mice? What would your hypothesis be? Do you think they got fat?
Less fat.
Less fat. Who thinks less fat? Who's just waiting to see the answer? Good. We've got a mixture. Honesty. I like it. So what they found is that these NAG mice have reduced weight gain. Even if they're on this Big Mac diet for almost 2 years, they don't get fat. Now everybody's thinking, so should I take ibuprofen all the time? Unfortunately, it doesn't work like that. So no, that is not a good response. They also found that they had increased longevity, or an increased lifespan. Whereas normal mice only live about 3 years, that's getting to be pretty old for mice in the lab, the nag mice can live out to 4 years. Even when they're on a high-fat diet, they have this increased longevity. So this was really interesting to the researchers, and people start thinking, well, we can just make a drug that acts in this way, and we can treat obesity. Unfortunately, there are other things that happen to these animals that we'll get into. They also found that the males were actually protected against colon cancer. So if they took mice and caused them to have colon cancer, the NAG1 mice did not get cancer as bad. They remained healthier. There's a study in humans that came out within the last year that showed men that had colon cancer and took NSAIDs or took ibuprofen consistently, they had reduced reoccurrence of the cancer later. And why that's happening is unknown. It's currently being studied. But so far we've just talked about males, or we've talked about what happens on a high-fat diet. And the title of this talk said we were going to talk about lactation. So what is the reproductive phenotype of the females? Right? That's where we're trying to get, is what's happening. And what happened is, at the NIH, where this work was done, they have a huge building. The entire basement is just mice. There's thousands of different types of mice that live in this basement. Each room that's holding the mice has a breeding technician. So for those of you that are students that are wondering, what kinds of jobs can I do? Large research institutions hire people to come in and help maintain and take care of these mice. And the breeding technician that was in the room where all of these nag mice were came up to the researchers one day and said, something weird is happening. The females don't have very many pups. There seems to be a difference in the number of animals that the nag females have compared to the control females. And so that caused the researcher that developed these nag mice to walk down the hallway to Dr. Korak's lab, which is who I did my postdoc with. And Dr. Korak is a reproductive biologist. The researchers that made these nag mice study metabolism and obesity, which is why they did that obesity study with the nag mice. And he said, we think there's a reproductive phenotype. Do you have anybody in your lab that might want to study it? I had just joined the lab. I was the newbie. So I was said, hey, on the side, why don't you do this experiment and see what's happening with these NAG1 mice? So the first experiment that I wanted to set up was a fertility study, because everything that was done originally was made by observations, that there was no actual data to support it. So as a good scientist, we should always have data to support our experiments. What we did was took that control mouse, took that nag mouse, and bred them with a proven male stud. What that means is the male mouse had successfully impregnated a different female, so we knew that he was fertile. And we bred these animals for 4 litters. So we wanted to make sure that the animals not only were able to carry pups, but they were able to have them over time. Sometimes with mice, brand new breeders, first-time moms, aren't very good. And they don't understand how to take care of their babies. By the time they have another litter, a month later, because mice can have them every 21 to 28 days, they're better moms and they will usually keep them. So we didn't want to miss that first part and see that they were bad moms. We wanted to see if there was actually anything as we moved forward. So if we look at the data from the fertility study, what we see, this is the control mice and this is the nag mice. And these are the pups born per litter. They have the same number of mice as the control animals. So there doesn't seem to be anything wrong with their reproductive phenotype or the ability to get pregnant and have the babies. But what we noticed was that there was a big decrease in the number of pups that actually survived to when they could be weaned. This is at the time where you can take away the baby mice, and they can eat food and drink water on their own, so you would put them in a different cage. When I say weaned, that's what happened. We had less than 2 mice being weaned from the nag animals, where we had close to 4 with the control. You'll notice a few of these mice, these aren't the same numbers, because both control and the nag mice did lose some pups over time. What was interesting is the data showed us that if the pups survived the first 72 hours that they were born, they tended to survive till they could be weaned. If they didn't survive during that first 72 hours, there was a significant reduction in nag mice compared to the control animals. So something was happening in that early time period for these pups that were born, because the moms could have them. Something was lost within it. So after the animals have these babies, one of the things that they need to do is they need to be able to lactate or be able to feed their pups. So because we're seeing this reduction in those first 72 hours, this led me to ask the question, well, what's happening within the mammary gland? Is there a difference in these animals? So the first thing we did was just look at the weight. While lactating, the mammary gland gets larger. It's making milk. It should be getting larger because it's holding that milk in. And what we saw was that the nag mice had smaller mammary glands at the time that they were lactating. So this suggested maybe they weren't making enough milk as it went through. So another thing that's very useful that you can do in mice is the animals are born almost transparent. We would call them pinkies. And those of you that have worked in the animal room here on campus, these are the young mice that sometimes get fed to our snakes and various creatures. Because they're transparent, you can see their stomachs. So you can actually look at the animal and say, do they have milk in their stomachs? Shown here as an example of a mouse pup on its side. It's 2 days old. And you can see that white spot there. That's actual milk in its stomach. So our breeding techs, we did a qualitative analysis where 1 corresponded to no milk spot present. 3 was a large, full stomach. 2 would be like a half-full stomach. And the question was, the mammary glands are smaller, but are the pups actually getting any milk from them? And when we look at those first 3 days, so when we saw that decreased survival, we also saw a decrease in the amount of milk that the pups had in their stomach. So this suggested to us the mice might not be getting as much milk as they need. If they're not getting enough milk, that's their nutrition, that's their energy. If they're not getting that food and nutrition that they need, maybe that's why they're not surviving. So because we saw this reduction in milk, we wanted to then ask, what is actually happening to the mammary gland? So the mammary gland is a very interesting tissue because it's constantly going through developmental changes. So when some people think of development, you think of everything that's happening while someone's pregnant, developing of the organism to make the organism. However, development continues after life as well. We go through a stage of puberty where hormones kick in and we begin to develop tissues differently. I think this post-embryonic development is most easily seen in frogs, which are born as tadpoles, and then go through a metamorphosis to be an adult frog. Humans and other species are going through similar type changes where hormones might be affecting these differences within them. And the question is, what is happening at the level of the mammary gland? So we're going to get into a little bit of development in the mammary gland. Males and females both have a mammary gland. This is Breast Cancer Awareness Month. Males and females can both get breast cancer. What happens is the mammary gland stays undeveloped until the time of puberty. At puberty, females begin making a hormone called estrogen. as well as a hormone called progesterone. And what that does is that stimulates the mammary glands to grow. By the time the female becomes an adult, you see that there's this large branching structure within the mammary gland that kind of looks like a tree, which is why we would call these branches as they're moving through. Males, even though they're born with a mammary gland, don't get these hormones on board, so you don't see this further development within the mammary gland. Although if they're given high doses of estrogen, you can begin to see some of these changes. During pregnancy, the mammary gland continues to develop. So when a woman's pregnant, you begin to get this growth, and these buds are forming over time. Those buds are what's going to help produce the milk during lactation. During lactation, you can see those small buds grow much larger. They actually, in some ways, kind of look like a bunch of grapes, if you think about it. And each one of those grapes is going to be filled with milk that can be secreted to feed the young over time. Shown here at the bottom is an example of a mouse mammary gland at the various different stages of development. So you can see the lymph node, and you can see that at this puberty, this is starting to grow out and you're getting that branching. You can see up here, there's not complete branching yet. By the time the mouse is an adult that has not been bred, you see this large branching structure moving out. During pregnancy, you begin to get these buds forming, and then at lactation, those buds are all filled in and begin to look kind of like bunches of grapes. So we saw that the mice were getting less milk, and we saw that the mammary glands were smaller. So what did the actual mammary glands of the nag mice look like? That was our next question. And if we look at a virgin mouse that had not been bred, you can see the lymph node is present here, and you can see branching in the wild type or control, as well as in the nag mice. So the mammary gland is developing like it should, but this isn't looking at pregnancy. So what happens during pregnancy? What we have here is the images across the top with an increase here at the bottom of the same mammary gland. And what we saw is that the wild type animals, they got these buds, they got those big bunches of grapes that were able to develop. With the nag animals, some of the mice developed normally and looked the same as the control. So there was no difference between them. However, in some of the animals, they never actually went through that final stage of development where those buds filled with milk and they looked like bunches of grapes. So we saw this small change over time. This led us to come to the conclusion that the mammary gland development was altered, although since it's not in all of the mice, we don't think that that was what was causing the complete change that we saw. So the next thing we looked at was the actual mammary gland tissue. This is an H&E section, and I think that you would agree that this, wild type or the control, does not look like this. Can everyone agree with me? What normally happens during development is you get these big circular secretory cells that are lining up. You can see these cells here. That is the milk that's present within that mammary gland. It's filling up the entire space within the mammary gland within those cells. Whereas in the nag mice, you see a lot of empty space. They're not getting large and milk-filled. They're staying really small. So they're like trapped in an early stage of development over time. So these mammary glands are not responding like we would normally see during pregnancy and during lactation, which suggests that nag, having that high level of that MSET-activated gene, might be affecting the ability of those mice to feed their young. So far, we've seen that these females have a reduced reproductive capacity. The pups have less milk than they get compared to the control animals. They have a reduced survival, and that the mammary gland is developed. But at this point, all data isn't actually looking at the process of lactation, it's looking at downstream effects. So the next question that I was tasked with answering is, well, is it actually the lactation that is affected? Right? Being able to produce milk and release it from the moms is really important for those animals to survive. So the first experiment that we did is called a lactation challenge. And this was a really fun experiment just to say I'm doing a lactation challenge, because everybody says, what does that mean? Right? Lactation, is it like, who knows what that means? There's so many options that we could go with, right? What this essentially is a measure of the rate of lactation. So in humans, after babies are born, what they'll do is measure their weight over time. And they'll say, are they gaining weight? Are they losing weight? Right? This can be used as a measure of lactation, because you're not going to take a human baby and look inside its stomach to see what's there. Not good.
They're not translucent.
They're not translucent. We can't look at those spots on them, right? So you have to use weight as a measure for how much milk they're getting. We did the same thing with these mice, where we took control or nag mice that were lactating. We separated the pups for a few hours. Put them on a little heating pad in a little corner. You play them a little lullaby, and they take a nap for a few hours. And then what you do is you come back, you take the pups, you weigh them, you add them back to their mom. So you have a starting weight for these animals. Once the mom has begun nursing them, once they're all attached to her and actively nursing, you start a timer. And you give those mice 30 minutes to eat their meal. After that, you weigh the pups again. That change in weight is going to be a measure of how much milk the mom was able to feed them, or how much milk the pups were able to eat during that time. If we look at the weight change between the control moms and the nag moms, we saw that these pups that were nursing from the nag mouse gained less weight. So this suggests that they're getting less milk. Even though they were all suckling or nursing the entire time, that whole 30 minutes. We actually sat and watched them, and if they stopped or if the mom moved away for a minute, we would watch her. These weren't animals where the mom was neglecting them, or she wasn't over in the corner having some food and ignoring her babies. She was trying to feed them. So it wasn't a behavioral problem. It was that they're getting less milk. So they're getting less milk, but that doesn't really tell us about the quality of the milk, right? I told you I was going to talk about ibuprofen and lactation. So the next question that we asked is, what does the milk look like in these mice? To do that, we took a mouse, we removed the pups, again for 3 hours. This allows the mom to build up milk. We then collected milk from a mouse. You can milk a mouse. Not as easy as milking a cat or other animals. Those of you that may have seen, some of you know which movie I'm talking about, right? So to milk the mouse, it's not like we can take cattle, hook them up to a machine, and use a machine to do it. The mice are too small for that. So what we actually did was we anesthetized the mouse, and you have to manually get milk, and then you have to have a partner that works in the lab that's willing to play the game of catching the milk as soon as it's released from the mammary gland with a pipette tip. Okay, so this was no small feat in being able to do this. I can tell you, I would be okay if I never had to milk a mouse again. I'm okay with it. But we were able to get milk. 50 microliters on average. 100 microliters was a good mom, and she gave us a lot of milk. So what we did is we took that milk and we actually wanted to analyze it and see what was present in the milk. What's different, right? The first thing we looked for, because this NAG1 gene is secreted, it can go into the bloodstream. The question was, is it in the milk? The control mice have no NAG1. They have no NAG in their milk, which is good because they don't have that gene. The NAG mice have really high levels in their milk. So this protein, NAG1, that's high in these mice, is actually being secreted into the milk that those mice are drinking. Okay? The other things that we looked at are non-steroidal fatty acids and triglycerides. So most of milk, especially for young mice, for young humans, is a really high fat percentage because that gives them the energy that they need for development over time. What we see is that there's actually a decrease of some of these fats in the milk. So in some ways, it's like low-fat milk that these mice are producing, rather than the high-fat milk that the control animals should be producing over time. So the last question that we asked is, okay, well, the milk has reduced fat, the milk has NAG in it, but is it actually affecting the animals at all? And to do this, we looked at endpoints in the serum. So just like when you go into the doctor for a blood test in your annual physical, they take some blood from you, and they're going to look at cholesterol and triglycerides and different readouts in the blood. We did this with the mice. And what we found in animals that were 9 days old, they had a lower level of triglycerides, which is their primary energy source at the time that they're growing. If we wait until these animals, and I should mention, the blue here are the males, and the pink are the females. So it didn't matter what sex the pups were, they all had this reduction at day 9. At day 21, this is at a time where the pups are ready to be weaned. They're mostly eating chow, whatever chow is in their food, but they're getting a little bit of milk. They're having a reduction in the males, although the females didn't show a significant difference. It should be noted we only had 4 females in each group, so that made some of this a little bit challenging. If we would have increased the numbers, we might have seen these differences. But really, this 9-day-old, where they're only getting the milk, I think is the most important one to think about, because these animals are getting less fat from the milk, which is going to give them less energy to go through and grow and develop. So the final question is, what is actually happening in the mammary gland? Why is NAG acting like this? So we took control mammary gland and NAG mammary gland. We did that same microarray analysis to look at different genes. Sometimes when you do this, you'll get a difference of 10,000 genes being different, and then you say, what do you do with 10,000 genes that are different? Right? That's a lot. I would lose track at like 2,000 if I tried to go through the list and look at them all. With this, we only found 289 genes that were different. 289 out of potential 25,000 different genes that we might have. We had a similar number that had higher and lower expression. So 289 is still a lot to go through and try to understand. I will tell you that the top 40 genes, top 20 genes, had never before been shown to be involved in mammary gland development at the time of lactation. So in some ways, that makes it even more challenging to do the experiment because there's no other literature that suggests this might be what's happening. So then it becomes, where do I go from here? I did this experiment after I had left NIEHS. So I couldn't even run down the hallway to the people that made the mice and say, what should I do? Do you know any of these genes? I have to email them and then wait for them to respond, wait for them to go through these processes, which sometimes takes a while. So what I did is use a program called Ingenuity Pathway Analysis, IPA. Very confusing because that's also my favorite type of beer. And what this does is it's this huge computer system where you can put in all of those genes that are different, and it creates networks and says these might be connected. So you get something that looks like this. Still doesn't really tell you a whole lot, right? Hard to say. Still just a bunch of lines, green, red, one goes up, one goes down. Still doesn't tell us what's happening within them. But what's unique about this is this pathway can say a lot of genes in this type of network are happening. So I mentioned earlier that I thought the milk was like a low-fat milk, right? Instead of a high-fat milk. One of the functions that comes up is lipid metabolism. Genes that are involved in making lipids. Genes that are involved in making fat. The nag mice have the altered ability to make fat. So these genes are currently being studied to understand what they might be doing in the mice. So that brings us back to humans. Everything I've told you so far is in a mouse. Well, how does that relate to humans? In humans, ibuprofen is fairly safe for prolonged use. Doctors actually tell women that are breastfeeding that ibuprofen is safe to use. There's a study that came out that shows that there's only about 1% of ibuprofen in the milk after a mother who is breastfeeding or actively lactating takes NSAIDs. There's 1% in the milk. That works out to a pediatric dose of about 0.02%. Seems safe, right? They're not getting anything. Donors that donate milk to milk banks, which is becoming a larger use, especially for infants that are in the NICU or premature babies, it's healthier for them to have milk versus formula. So the donors that produce milk and provide it for these milk banks for the different hospitals are told that it's safe if they take any ibuprofen. They don't want them taking other types of narcotics and drugs. But it's safe for them to use this milk. None of these studies looked at whether this NAG gene is in the milk. We know that humans make NAG1 after they take NSAID, although all those studies have been done in males. We don't know what happens in females. So from this, we can conclude that these NAG mice have altered NAG levels in the milk, altered growth. Ibuprofens are safe for humans. What does this mean in terms of a human? We're not going to all run out and take ibuprofen in order to control our obesity or in order to do things. What I think it means is really there's always more research to do. In science, you'll see that once you finish one experiment, you usually have 2 or 3 more questions that you're going to ask as you move through. And these studies now are suggesting that we shouldn't only look at the active drug that somebody's taking, especially for breastfeeding women, but we should consider what other byproducts might happen. So we know in human males that take NSAIDs, NAG1 gets increased. We haven't looked in females or in the milk of females that is actively breastfeeding. So that might be a good start, right? Let's do some experiments where we actually see, is this gene being secreted into the milk? We don't know if it's altered by them. And this suggests that a lot of clinical endpoints that we do in drug design, they give somebody the drug, they look for that drug. Especially with lactation. They give the female the drug, they look in the milk. What they're not looking for is other metabolites that might break down. Other proteins that might be present, such as NAG1 within the milk. So at this point, I think the take-home would be take drugs at your own risk and think about what might be happening, right? Don't act like these NAG mice and take really high levels over time. These animals aren't just having an occasional injury where they're taking it, but they're taking these drugs consistently. So the end game is it's happening in the mice. Where do we go from here? So with that, I need to thank my advisor while I was a postdoc in Korat. Tom Eling's lab is the lab that developed these mice, and he's the one that came down the hallway that one day and set me on a path of milking mice, which I never thought I would do. Our Our researchers Justin and Janardon, because I've never been able to say his first name, and he lets me call him Janardon because he said nobody can say his first name. And Glenda Moser these are all co-authors on this manuscript. She's the technician that worked in that breeding lab that made the original observation and was involved in the study throughout. So for those of you students that are thinking about, well, if we're just taking care of the mice, you can contribute to the research, to the studies that's going on, and there are these great potentials. So with that, I need to thank my department, all of the minions, and I'll take any questions.